Hello students, welcome to Vedantu Neat English. Hi students, welcome to Vedantu Neat English. This is your teacher Diksha Kaushal and this is
Gopika ma'am.
and we are here with one more teacher. Can you guess who is that?
Exactly.
That is there in the thumbnail.
What is the plan today, ma'am? What are you guys doing?
So, ma'am, we have, as you know, we have biomolecules in bio as well and chemistry as well. So, basically, it's a fusion of bio and chemistry in biomolecules.
Biochem.
So, we will do some questions, right, that can be asked in your NEET exam, some PYQs, and with that, we will see what is this, what are the things that are similar, and how with the help of chemistry and biomolecules, you can together gain around, I think, in bio also 3 to 4 questions, in
Yeah.
chemistry also 3 to 4 questions. So total 7-8 questions you can think about.
Okay, awesome. So I am the guest of honor today. I came to cut the ribbons
chief
of the
guest.
class.
Chief guest.
Okay, Basar sir, can you please show your face?
Yes, ma'am. Yes,
So
ma'am.
then they know.
Hello. Yes, sir.
Shreya sir is also here.
Yes, sir. It's live, sir.
So full
Yes.
day is
Oh
here.
my God. Man, this is like crazy. Some dystopian RNA, DNA stuff. Oh my
Yeah.
God, crazy. Hi, guys. Welcome aboard.
Hello.
Yep.
Then the chats are still loading. Huh?
Yo.
Yeah.
Green and red. Green for Baswa, sir. Red for Diksha, ma'am.
So today is basically about DNA, RNA, sir, only. Our
Okay.
batch's name, and it's from biomolecules. So we are going to discuss biomolecules only.
Awesome.
DNA, RNA.
Cool. So all the best, guys, and support your favorite teacher. I hope, yeah, it should be the ones who are not going to be in the class. Somebody who is outside.
Exactly.
Yeah, we'll be in the chats. Okay. Chalo. Take care, guys. All the best. Bye.
Okay, bye bye, guys.
Bye-bye.
So now let us start this. And so, guys, tell me, do you like biomolecules or do you like chemistry biomolecules or more or biomolecules more?
Yes, students. That's the first question to all of you. Which one is better, or which one do you like more, the biology or the chemistry? All of you know biology is better.
No, chemistry is better. Chemistry is the best. If you don't have chemistry in bio, also you are studying all the structures and hydrogen bonding. If you don't know chemistry, how you will be able to study that?
Ma'am, I didn't say which. I said chemistry you should know, but I said which is better
Definitely.
because we
chemistry
can
is
relate to
better.
biology more. It is happening inside our body.
And the reactions are also happening inside you, like
Uh-huh.
photosynthesis, the
Exactly.
digestion, everything. So basically, bio and chemistry are like
Oh yeah, friends.
friends.
Okay, like same,
Okay.
same but different.
Same but different.
Yes. So, with that being said, students, let's start today's class. We have around good questions are there. So let's solve the questions and let's see which you're able to solve. And
So-
the small twist is gonna be we will try to solve each other's question also, right, ma'am?
Yeah, that will be amazing, no?
Will be nice. That will be
try.
nice. Okay.
Okay, so let us start this.
The first question.
First question, that is my question, chemistry question. So, guys, do not disappoint me, right? Do not disappoint me. So, first question is, do you want to
I'll
read
read
it
it.
out?
RNA is different from DNA because RNA contains. RNA is U2 batch, DNA is DNA batch.
Yeah, that's what is exactly that? So, I think I'm feeling so weird with these heels. Yeah, now it's better. Every student think that I'm really tall, but actually, when I remove my heels, they're like, "Oh, ma'am, you're not that tall." So, RNA, yeah, RNA is basically
different from DNA because RNA contains.
RNA
Okay.
contains.
This is a little easy question, right, friends? This is a little easy question, not one. Look at option number 2, all of you. Look at option number 2. Here it says ribose, sugar, and uracil. So uracil is in the case of your RNA, ATGC in the case of DNA. Easy
Yes,
question.
absolutely right, sir. I think that is exactly there in your bio part.
Yes, ma'am. So students, Diksha sir and Diksha ma'am on the screen.
Yeah, oh really? You know, I've been getting these messages that whenever I'm there, especially in DNA batch, I'm there, na, students are like, hello Dikshant sir. I'm like, who is this Dikshant sir? Do I look like a man now? What's your problem? Now I got to know actually
Yes, that doesn't
this
mean that.
is who knows about Diksha sir and Baswa ma'am.
Baswa ma'am. Diksha sir and Baswa ma'am.
The names are interchanged, is it?
So this was an easy question, right, ma'am? Easy question, ATGC.
ATGC.
Okay. This is a good question for you. Bio question. Students, this is your NEET 2024 PYQ. Only 77% of students were able to answer this question. Many students went wrong here. So let's see who is able to answer this question. All of you.
Okay. So, which of the following statement is incorrect about enzymes? Okay, so they are highly substrate. So I know some of the points here. I don't know the correct answer because I can clearly see that is not there in biomolecules in chemistry. So enzymes are basically biocatalysts, right?
Biocatalysts,
So
yeah.
catalyst can be positive or negative. They are yeah, they can enhance the rate of
reaction or
decrease. When it decreases, they are called inhibitors. All the things that I know, I will tell. Acha, it is same. Okay. So, okay, inhibitors is poison that we call in chemistry, we call
Chemistry,
it poison,
poison.
right? And positive catalyst, you call it
As promoter, I just say promoter.
promoters. Okay. So, they're highly substrate. Substrate is that reactant,
Huh.
right?
So they're asking incorrect, ma'am. So the question is which of the
I
following
think
is
I know the answer.
incorrect?
I think I know the answer. I think this is true
Huh,
because they are highly substrate. The thermophilic. Thermo means heat. Philic means love.
Love it.
So thermophilic organism enzyme can catalyze reaction at high temperature. So at high temperature, the rate of we know in chemical equilibrium that
Now I lost it. Chemical equilibrium and all. Chemical
when you
equilibrium.
when you increase that, but that depends on whether it's endothermic or exothermic reaction.
Ma'am, I'll give you a free pass. Option number 2 is true.
So that is true. Okay.
So
Like
let me tell you, it's a true statement because there is something called as Taq polymerase. Taq polymerase is an enzyme which is derived from a Thermus aquaticus, which is the organism staying in acidic conditions. So it can work at 9 degrees Celsius.
okay, I wish I was that organism. No, reason is
Okay.
you know the reason I told you morning, no? All enzymes are
Proteinaceous in nature.
proteinaceous in nature. Okay, means they have proteins.
So basically, enzymes and proteins are same to same.
Okay.
Enzymes are made up of protein, but here it says all enzymes. That is the clue.
That's false.
Why?
I just thought the way you are saying it. Maybe there are some enzymes that does not have protein.
Exactly. There
Right?
is something called as ribozymes. They're RNA enzymes.
Oh yeah, ribozymes.
ribozyme.
Yeah.
Exactly,
okay.
ribozyme.
Third is the correct answer.
Third is the correct answer. Students, how many of you are able to answer this question? How many of you are able to answer this question correctly? Ribozyme. Tell me in the chat, all of you. Ribozymes. Quickly.
I'm not a cheater because I
Yeah,
am
I saw. Ma'am is not a cheater.
Dedicated person.
Okay. Students, easy. Right? No, it says all, yes, or it's not all. Next question for me, right?
So, the pyrimidine bases present in DNA are.
Okay, this is again, ma'am has given deliberately easy questions to me, thinking that we will not know anything. So, students, this is again an easy question. All of you know there is something called as purines and pyrimidines. So, the way I learned it, right, ma'am? Where is the pen? One second. That doesn't erase like that. So, cut pyramid. Cut the pyramid. Cut the pyramid. That is going to be your cytosine, thymidine. Cut the pyrimidines.
And purine is in AG.
AG.
AG, OG, LOG, SNOG.
Yes, that only.
That's how I
You
remember.
remember it?
things.
Huh.
Yeah, I remember. See, you can't cram everything, right? So
true.
you have to have
Some tricks.
some tricks. That is important. Otherwise,
yeah.
we'll go mad.
Mental. I said mental.
Mental. Yeah.
Mental.
Psycho.
Okay, so here cytosine.
Cytosine and thymine.
Yeah.
This becomes the correct answer.
Right.
Third is the correct
How
answer.
many of you are able to answer the question very clearly?
Very good.
Easy question again, right? Cut the pyramid. All of you remember? Cut the pyramid. So cytosine, uracil, thymidine are gonna be your pyrimidines.
Very good. Very good. Very good. So next is yes, guys, smash the like button if
Okay.
you're liking this video. If you want more sessions like this, then do also share this with your friends, right? So that they can also join here.
Okay, next
So-
question for you, ma'am. Again, it's a NEET 2024 question. Okay, question. Easy
Okay, that's a easy question. Zwitterion. The following can be found as a zwitterion, fatty acid, monosaccharide, amino. That's such a simple question. Amino acid, right? So what is a zwitterion?
Ma'am, you explain them. It's
Yeah,
very easy question.
I need to write it. Yeah.
Sorry, took the pen away.
Okay, so I'll write it. So basically, zwitterion is, we know that it has amino acid, has one acidic part, that is COOH, and I'm taking just R, any simple chain I am taking, just R I am taking. This is your NH2, right? So this NH2, this is your basic part and this is your acidic part. We know that what is the work of base? Base will it will accept the H positive. COH, acidic part will donate H positive. So what will happen?
H3CO minus, right?
Yeah, so it will donate its proton, and this will become like, this is NH3, this will positive, because when nitrogen is forming 4 bonds, so it will have positive
Positive,
charge. So I have a trick for this. Oxygen forms always 2 bonds, nitrogen always form 3 bonds. So for example, you're expecting 90 marks, then you suddenly get 99, you will be happy, na? Positive. So it is supposed to form 3. No, it is forming 4. It is very happy.
loss, so
So it will be negative. Yeah.
simple.
So sad, na? You lost.
So student, this is called as a Zwitter ion. It is called as a Zwit.
Anything you can
Put
say here. Or you can say anything.
everything gone.
Yeah.
So student, this is a Zwitter ion. All of you know Zwitter ion has a net zero. Zwitter ion has a net negative neutral charge because, ma'am, neutral charge. Why? Because it is cancelling
Yeah,
out,
because
right?
it is cancelling out each other. So all over it is a neutral charge, right? So
easy question,
it
actually.
was
Very-
very
neat
easy.
PYQ.
NEET 2024.
This year
Also,
only.
simple questions can be asked, right?
Oh.
A secondary structure. This though I expect, hai na? I think that is there in your.
Yeah, so students, all of you know, right? There is something called as primary structure, secondary structure, tertiary structure, quaternary structure. In primary structure, it is a linear chain of amino acids, only and only peptide bond. In secondary structure, we have two things, that is alpha helix and beta plated. In tertiary structure, we have the ball, round woolen ball with 3D in shape, and quaternary, the hemoglobin one example. So here they have given you a secondary structure of protein, that is going to be your alpha helix backbone, right?
Simple.
Very simple. It's a very simple question.
Next question. Hydrophobic interaction sequence. This though options are also not that confusing. Simple answer. Okay,
This is
sir.
a good
This one
question.
is
This is a very good question. Let's see how many of you are able to answer this question. All of you in the board, students, quickly smash the like button. All of you, share this video with everyone, and let us know in the comment section of today's video if you want more such videos which are fun and also interactive and learning happen.
informative. Yeah.
Exactly, ma'am.
So
So let's see if we can solve this question now.
my brain is totally not. My brain is
is
not
not braining.
braining.
This is a good question.
Okay, I'll use some guesswork here
Magic.
because I have no idea about
Inky,
this.
pinky, ponky.
Inky Pinky Ponky. So
Okay, students, while ma'am is answering the question, all of you tell me the answer in the chat. All of you quickly.
okay, I will not see that answer. Okay, I will not. I'm not a cheater. I'm a very loyal person. Which of the following graph explains the effect of pH on the enzyme? Can you tell me a little bit about enzyme activity?
So students, a little ma'am also. Every single enzyme is very picky. Ma'am, do you eat all the food in the world? You picky, eat a little bit
Yeah,
picky.
you know that.
So enzymes are also very picky. They're like, I need a proper temperature, I need a proper pH, and we call that optimum temperature and optimum pH. So here you need to understand, point out the graph which is showing a optimal temperature at where a P or optimum pH where enzyme is working. For example, ma'am, in your mouth you have salivary amylase enzyme. So though that enzyme will work specifically at one pH.
Right.
So if pH is going this side, it doesn't work.
Even our digestive juices will work at a proper.
Every single enzyme, ma'am, every single enzyme will work at a very specific pH. Now you need to tell me which one looks optimum here.
Okay.
I'll
So
give you a small clue, ma'am. These two are not there. It's either this or this.
what's the difference?
Here x
Acha,
axis
acha.
y axis. That's all. That's all the difference.
Okay, so let me see. Here we are saying that when enzyme activity is increasing as the pH is increasing, that means it is going towards a basic condition, right?
But it's
And then
coming
to
down
one
again.
it is maximum. Then after that maxima, it is decreasing. Maybe after a certain amount of, it has reached a certain amount of basicity, it will decrease. I think this can be the correct answer. Wait a minute, let me see this also. I think this is the correct answer.
Which one, ma'am?
2.
Ma'am, this is a direct question which will tell you the only difference is going to be pH here. This is the image of NCRT which directly tells you. pH should be always in the x-axis and enzyme activity should be on the y-axis. That's all. So it is a very minute thing where many students can go wrong.
Yeah, that's so true. It's because it's confusing, right? Students can assume that, okay, ma'am, maybe enzyme activity is here. The reason for dipole helical structure of DNA is?
Okay, the reason for double helical structure of dipole, no? It's a dipole. Double helical structure of DNA is operation of. Okay, this is again easy question. This also is a very easy question. Again, all of you know structure of DNA, right? Structure of DNA, all of you know, right? If you're part of DNA, you should know. And in DNA, there's something called as hydrogen bonding. Yes. So hydrogen bonding is the answer, right?
Yeah. So as you know in DNA, है ना, we know that our DNA batch students also they know we have hydrogen bonding, right? So basically, sir, you are saying hydrogen bonding. That is absolutely correct. But I want to know what exactly do you think about what is the difference between dipole-dipole interaction, electrostatic interaction, van der Waals forces, hydrogen. Okay. So now, any of the definition that you can tell us.
Okay, I don't remember any of them, but I remember van der Waals forces are weak forces, right?
Yeah.
They are weak
right,
forces.
Right.
And electrostatic, no idea at all, don't remember. Dipole-dipole interaction, I remember it is a partial positive and partial negative charge.
Very nice, sir. Very
That's
nice.
all I remember.
I'm
That's all.
impressed. Very
That's
nice.
all I remember. Dipole-dipole I remember because we used it a lot in biology also.
Hmm.
Electrostatic and van der Waals I don't remember much.
Okay, so basically dipole-dipole interactions are, for example, this is A, this is B, right? I am saying that A is very clever. So it will try to pull out the electrons from this B. So it will have a partial negative and partial positive charge. So there's also another molecule of AB which is partially negative and this is positive. So now these two are going to have, because of the polarity that they have formed here. One is negative charge. Because of the, here the electrons are more towards the A part, right? So because of this, they don't have exactly a negative and positive charge, but they have developed a polarity. Because of that, it is called dipole. Dipole interaction. Partial positive. That was right. And electrostatic is nothing but it's like salt, like NaCl. Electrostatic interactions are ionic.
Ionic in nature.
Ionic,
Ionic,
yeah. Van der Waals forces, you were right, it are weak forces, but it is between two molecules.
Okay.
But these molecules need not have any dipole, any polarity difference. For example, H2 and H2. The interactions that is existing between them, right? That is called the Van der Waals
Weak
forces.
forces between the two
Very
molecules
weak, weakest forces, I would say.
Can you
सबसे
break
ज़्यादा.
it? Can you pull it apart?
Not even pull it apart. They are like you are standing there, I am standing
So we
there.
have Van der Waals
So it's
forces
like that interaction. For example, in the exam hall, we both are sitting, है ना? I don't know some question you want me to. You are answering me. How you will answer me? Like 1, 2, 3 for ऐसा. You'll answer me. That is van der Waals. We are not talking to each other ऐसा. We are interacting with each other like that.
Okay.
Yeah. Okay. Hydrogen bonding, so we all know that this is one hydrogen, this is A. And so, Risa, tell me one thing. If this is hydrogen and this is A, this is not electronegative. For example, this is, I'm saying, not electronegative.
Students, honestly, I'm going somewhere now. Chemistry I learned today.
I'm like, sir has given me such difficult questions. Now I will take the revenge. I will confuse him.
So you're saying something.
So this is B. I am
Okay.
saying that, okay. Let's say one thing. Okay, I'm giving you one example here. We know what are the electronegative elements FON, phone. Fluorine, oxygen, nitrogen.
Fluorine,
Fluorine, oxygen, nitrogen. Students really get confused, and that's why I'm asking for this one, so that the students who don't know this question, they can also learn something here, right?
Okay.
So FON, basically these are the most electronegative element, very clever element. They are, I want this money, give me these electron. Very
Okay.
clever.
Very clever.
Okay, very
Cunning,
clever.
cunning.
Phone, very cunning. So we call it phone. FON, phone.
You call phone, you're like, give me money.
Phone.
phone.
Yeah. So fluorine, oxygen, nitrogen. So this is your HF,
Okay.
and this is some. I would say this is maybe. Let us call it bromine.
Okay.
So there is hydrogen here.
Okay.
Will hydrogen bonding exist between them?
No.
Yeah.
Because it's
Why?
more electronegative in nature.
Actually, the reason here is you're slightly close here. The reason here is the hydrogen that is connected here should be connected to a electron negative element, FON, any of these, and the other element should also be electronegative. So
Okay.
here you will have no hydrogen bonding. For example, they have given you something like this, CH, and here they have given you oxygen.
So the hydrogen will go this side now.
Here also the hydrogen bonding will not happen because the hydrogen bonding is with that hydrogen which actually is directly linked to some electronegative element.
Okay.
Only then it can show.
So since it's with carbon doesn't there's no electronegative,
Electron-
not show bonding there.
Right. Why I
Beautiful.
told this question, because there is one very important question that is asked in organic chemistry, that you have this ether ROR, right? And you have ROH. Students say that this also has hydrogen bonding. This does not have.
hydrogen because there is no electronegative element there.
Yeah, Dekho, sir also answered it correctly, right? So
So,
you never forget this now.
Such good question, right? Such good
Yeah.
question. So, students, this is how beautiful ma'am taught me. I did not even know about it. Probably I read it in my class 11th. I forgot about it, but now I learned something new again. So nice.
So, next is.
Students, all of you are asking, will they be asking in your NEET examination? Yes, all the questions are 90% of them are NEET PYQ. Can you see here? This question right here is from your NEET 2023 and NCRT question. Let's see to the question.
So all the question, even chemistry, they're also picked from your PYQs only, right? Very important. So now I know something about this. Cellulose does not form blue color with iodine. Dekho, iodine. I know. What I know
Now,
about iodine.
I know for a fact that ma'am will not tell me the answer till teach me something extra now. I know it. Ma'am, continue. Continue.
I'll continue. Iodine, right?
One second, ma'am.
So iodine itself does not have any color, right? Iodine itself does not have any color. But if you give it starch, starch is acting as an indicator, right?
Okay.
So we usually say iodine is of blue color. It's not like iodine like itself gives blue color.
color.
With starch, there is also one titration. If my students, we have already completed that in redox reactions, if you remember that. Like whenever iodine sees starch, it gets excited. They're like, okay, I want to give blue color here.
Okay.
So iodine with starch, it is giving blue color. That I know.
Okay. Now, ma'am, I'll tell you the option. No, you tell the answer.
Cellulose does not form blue color with iodine because cellulose does not form blue color with iodine. Blue color with iodine. Maybe iodine is converted to I negative.
See, here you told starch is forming blue color. You told me starch is forming blue color. It means something starch has which is not present in cellulose. Right?
Okay, it is basically I know.
Yeah, you know it. Now you know it. I know you know it.
Okay, so starch and cellulose, the difference between starch and cellulose I have to tell, right? It is cellulose contain complex cellulose
Look at it. Because any
they have not
options-
shown
any
any
options talks about structure anywhere, ma'am.
structure only. I'm looking for the linkages actually. It does not contain complex helices and hence cannot hold iodine. Does not form blue colors. Cellulose does not form blue color. So I think fourth is the correct answer. Oh,
Absolutely. Absolutely. Ma'am, like in chemistry we learn, right? Your amylose and amylopectin are two parts of your starch.
Yeah.
Like
Amylose is alpha 1 4 linkages and amylopectin is alpha 1 6 linkages.
Beautiful. See,
Right.
so for example, I'll give you a small thing. Imagine you do not have fingers. If I give you this pen, will you be able to catch this pen? No. So that is the case with cellulose. Cellulose does not have alpha 6 linkage. Does not have alpha 6 linkage at all. So it cannot hold iodine. Now, whereas the cellulose does not have any arm, cannot hold at all. But starch, ma'am, starch has the alpha 1,6 linkage. It means it has Loeys fingers. It can hold iodine. That's all. So it does not contain complex helicals and cannot hold I2. That is, cellulose does not have any complex structures which cannot hold I2.
Got it.
So
Basically, it is based on the linkages.
Linkages.
That's a very nice.
Good question, no? Good question, no? Okay, next question.
Which of the following is non-reducing sugar? Which of the following is a non-reducing sugar?
Okay, now I'll tell you the bio definition which I know. Non-reducing sugars are sugars which does not have a free aldehyde or ketone group. Does not have
Carbonyl group.
carbonyl groups.
Right.
Okay, this is an easy question again, ma'am. You're giving me easy questions, not deliberately. Okay, students, all of you know, right? Your polysaccharides and disaccharides are non-reducing sugars. They are non-reducing sugars, right? They're non-reducing sugars. Reducing sugars. Following is a non-reducing sugar. is a
It's an exception.
it's an exception. That is yours. Sucrose.
Yes.
Sucrose, right? Sucrose, right? Sucrose. Sucrose is a disaccharide,
But it is not.
non-reducing sugar.
Right. Acha, one more thing here. Now, you said that no.
What happened now? Why are you laughing? I know I'm funny, but I'm not that funny also. What happened to ma'am? What happened, ma'am? Students, did you say something? Tell me. Did you say something?
I don't know why I'm laughing. I just remember something about reducing sugar.
What do you remember?
That's my when I was in my class.
Share us also. Share the laughter. What is happening?
I don't know. That was a very weird joke. I don't know why I laughed. My sir, the sir that told us about reducing non-reducing sugar, he told us about something like girlfriend, boyfriend, the girlfriend who are free are non-reducing,
Okay.
something like that. So that made me laugh. I know that's not funny, but
Honestly, honestly, I didn't understand the joke only. Ma'am, you keep laughing, ma'am. You keep laughing. It's fine. Keep laughing. It's fine. Students.
sometimes this happens to me. Sometimes they know that this happens. Sometimes.
But I didn't understand the joke at all. But I don't know what.
Sometimes out of nowhere I will laugh like that. It's a therapy, laughing therapy. I do.
It is also scary. Imagine in the night time, you suddenly start laughing. I will run away from you, ma'am. I will run away as fast as possible away from you.
Just imagine the lights are blinking. You know, in my class the board is always blinking. So just imagine the
We'll go to
board
the next
is
question. We'll go to the next question. Next.
I have to tell something to my kids.
Okay.
Wait, I'm serious now. Okay, so this non-reducing sugar. Okay, sir, tell me. I'll tell you. In chemistry, basically, a non-reducing sugar is that which is as a non-reducing, right? Which is not reducing sugar is that which reduces something.
Okay.
Basically, for example, I'm saying I'm a reducing agent, then I will reduce you. If I'm an oxidizing agent, I will oxidize you.
But itself will undergo opposite of it,
Ox-
right?
yeah.
So if I'm reducing agent, I will undergo oxidation.
Yeah, perfect. Kya baat. Very nice. So basically, the sugars that can reduce the other thing. For example, they can give Tollens reagent test. They can give Tollens, the silver mirror test.
Okay, don't remember.
It's very beautiful silver mirror on the tube it will form. Right. Basically, it will convert the Ag positive to Ag.
Negative.
Ag
Silver.
positive to Ag. Basically, we know that let's not get into it. Right. Basically, it is reducing the other thing.
things,
Right.
okay.
Okay. It is decreasing the oxidation number here. +1 to 0. It's
Yeah.
number is decreasing.
Yes, it is decreasing. So it is.
1 to 0 number is decreasing. When the number is decreasing, it is reduction is happening. When number is increasing, oxidation is happening.
So in what I usually teach is reduction is gain of electrons, of gain of protons.
Yeah.
Oxidation is loss. Oxidation is loss.
So here also, if you see 1 plus 1 and this is 0, you are saying reduction is gain of electrons. If I make it 1 electron will have -1 charge.
-1.
So
it's
if I
becoming positive.
Haan.
It's gaining.
So the simple definition is
Plus
next
one
time
plus.
you can teach the students
Next
that
time I'll use
reduction
this.
is oxidation number is decreasing and oxidation number is increasing.
We'll try it. Oof.
है ना?
So many concepts today. So many concepts.
So
I think my
many
brain is gonna fry now.
You know what I love about this chapter? You're telling something about this and
Exactly.
I'm telling something else about this.
But everything is connected.
Everything is connected to each other. That's the beauty of science.
Exactly. And students, in examination, if they ask you basically about reducing sugars, monosaccharides, right?
Acha, which of the following is not a secondary metabolite?
out of
Sir is like, I don't know which Revens. I think biomolecules is more of a bio chapter.
Yes, it's more
That's
of a bio.
why the concepts that are there in chemistry that are also there in bio.
Exactly.
Only
not on the ground.
Not the other way around. These kind of concepts are not there in NEET 2023 question it is. Which of the following is not a secondary
Students,
metabolite?
do you realize ma'am got such easy questions? I am shooting ma'am with PYQs.
And even from biomolecules, from chemistry, easy questions are asked. Yeah, those kind of questions are asked. Really, it's easy questions are asked from biomolecules.
Let me try this one question. Try this.
I have no idea. Oh no, I know about morphin.
Okay, I'll tell you what
morphin.
is secondary metabolite first of all.
Yeah.
So, plants produce a lot of substances. So when they produce certain substance, some substances are used by the plants in their life cycle,
Okay.
like fats, proteins, carbohydrates. They produce and they use it for themselves.
Right.
Now there is something called a secondary metabolite, which is produced in the plant's life, but it is not used by the plant.
Okay.
Now, do you like palau?
I love it.
In the palau, they will add that cinnamon, chakke, cinnamon.
Yeah, cinnamon. Yeah.
Huh, that is basically a type of bark or turmeric, or not turmeric. Then we have cough medicine you drink. It
Yeah.
has something called as codeine. That is and
Yeah, codeine.
codeine. Then there is morphine here. Morphine is added to patients. That is also derived from the plants.
You know, when I was in my master's, MSc second year, I had a full dedicated subject of natural products. So there I read all these structures, big,
Okay.
big structures,
Okay,
but
so imagine, did you learn about morphine there?
I learned the structure of morphine, but I now I
So
don't
you learned about anthocyanin.
this I
It is a pigment. Anthocyanin is
Okay.
pigment, plant based.
like apna cytoplasm.
No, cytoplasm.
Cytoplasm nahi chlorophyll.
Chlorophyll. Chlorophyll.
Cytoplasm is there in
Okay.
the cell, right?
Yes, curcumin. Curcumin is obtained from turmeric, curcumin longa. So curcumin, anthocyanin, morphine. All three are your plant secondary metabolites, but lecithin is not. Lecithin is a phospholipid.
Phospholipid means?
Phospholipids are lipids which are present in the cell wall. The cell wall.
Okay, cell wall.
It's a plasma membrane.
Okay, the plasma.
Cell wall.
membrane. Yeah.
Plasma.
Plants and animals, the difference between plants and an animal cell is that one has wall and
Ah,
one
so
doesn't.
plant has cell wall, animal cell does not have a cell wall.
I remember this from my 8th class lecture.
That was a good question. So this question will directly ask from here. Can you see here? This is a list of all the secondary metabolites. Can you see here? Carotenoids, anthocyanin. We saw there. It's a pigment. We learned about codeine. I told you just now. Morphine. It's an alkaloid.
Alkaloid, maybe it's basic in nature.
Exactly. Then we have terpenoids, monoterpenes, diterpenes. Then essential oils. You know that nice smell.
That used to skin also.
Exactly,
These,
Korean
yeah,
skin.
yeah. They say, okay, use this.
So lemongrass oil. Then there are toxins called as arbin and ricin. Then there is lectins called as concanavalin A. Drugs called as vinblastine, curcumin.
Pronouncing these words, it's so tough. My tongue will do yoga only with this. It's such a tongue twister. It is. You give someone bio book, it's a tongue twister.
Yeah, for non-bio students, it will be a little tongue twister.
Yeah, it's totally.
What are you telling? Ma'am will tell some IUPAC nomenclature 1,6 blah, 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 I'll go.
That's so simple, but this is difficult. Wind blasted.
Wind blasted. No, ma'am, do you know, have you seen this pretty pink color flowers on the roadside?
Yeah.
So it is derived from the pink color flowers, and this particular vinblastine has cancer curing property.
Ooh, like cisplatin.
Yeah. So there's something called vinblastine and crisp. I think that was the one you told.
Cisplatin.
Curcumin is from drugs, then rubber, gum, cellulose.
Cellulose,
All
the polymeric substances.
So all of these are secondary metabolites. So in examination, they will directly ask secondary metabolites, and they will ask you which one is not. See,
This is from NCRT.
NCRT snip. See,
This is
so
important.
lecithin is not a secondary metabolites. So they have
NCRT is so very important for bio, right?
exactly direct question. Okay. I think I saw the answer, ma'am. I think I saw the answer.
Okay.
Okay. Which of the following is amino acid is neutral? Again, easy question, ma'am. Answer is given here. That is glycine is given here.
Aspartic is
Acidic,
and
isin and argine are basic in nature. So all of you remember the structure of glycine, right, students? That's going to be your COH,
and the CH2.
NH2 and H and H.
To every chemistry teacher, when someone is writing as a C, it hurts.
Why?
Why you have not completed the tetravalency of carbon? Why this two carbon? I usually, in chemistry, so usually we can write it like this also, right? Like this also, right? But I have this obsession
Okay.
of writing because we know carbon is making bond, right? So I have this obsession of writing it like
carbon
this.
only should come. From
Yeah.
carbon only should arise, right?
Yeah, for me, it's that obsession. Students have observed that in the class as well.
Okay.
That ma'am has this obsession of right bond.
Right
should be
bond
there.
should there. This should bond here only. from where something else should not come.
Yeah, I'm also like, why you on homolytic cleavage? Why you are not making the bond like this? Why you are making the bond like this?
Ma'am, I remember just now one more thing. How many of you know what is a chiral carbon? Shoot.
Yeah, chiral carbon is very important. I
Uh,
love chiral carbon.
So, students, do you realize here the glycine is the only amino acid which is non-chiral in non-chiral in nature because it is all four are not same. Right? It is the
Yeah, so basically in chiral thing, there are two conditions. One is it should be that thing that we're looking, whether you're talking about carbon or any other bond,
carbon, right?
it should be, yeah. Alpha carbon means, for example, I'm talking about this carbon. It should be sp3 hybridized. Basically, it should have four bonds, four single bonds should be there. If I'm talking about it has this kind of structure, one double bond, then you're saying this is A bond is attached here, B here, D is attached, then it's not a chiral carbon. You should have,
four separate function groups should be there, right?
yeah, you like you have studied it by four separate function groups. So, but basic the core chemistry language we say it should be sp3 hybridized. Basically,
Okay, I
it should
don't
have
remember this.
four sigma bonds.
Four sigma bonds.
Sigma bonds. So in this sigma
bond.
sigma bond.
Okay,
Yeah, not a cutie pie bond should be there.
except
The
sigma
pi
bond.
bond is a cute bond. Sigma bond is like
chat
sure,
bond.
like very, very swaggy bond,
Okay.
right? So sigma bond should be there. No, pi bond should be there.
Okay, ma'am. What is the difference between sigma and pi bond, though?
So basically,
Is it
very
going too long?
interesting. It is very interesting.
Okay.
And for example, I'm coming here, you're coming here. Okay.
Okay.
Right. So we are shaking hands.
Okay.
It's axial, right? It's
Axial. Axial.
So it is the head-on collision.
Okay.
For example, you're coming here and it's head-on
Both headers
collision.
banging.
So, right, that will be head-on collision.
Collision.
So in that case, sigma bond we are making, right?
Okay.
So for example, it's this. These are electrons. These are other electrons.
Okay.
This is one electrons mean this is one orbital.
Okay.
This is another orbital.
Okay.
They are not ऐसा colliding
Head-on
with each
collision.
other. They are
They're not-
colliding with, like for example, this is its slope. This is its slope. They are now sharing it like this.
Directly. So it is joining somewhere like that you told, right?
Yeah. So basically, it's like this.
Okay. This is
So
Q
this is
pi.
That is why it is so loose.
Okay.
That is why it is difficult to break sigma bonds, but pi bond is easily it can be break.
Because pi bonds are arranged in a this.
You
Okay.
have you studied resonance?
Yeah, long back. I still remember. So crystal field theory and valence bond theory. Two favorite things. That's all I remember.
Yeah, they are interesting.
Oh God.
So that was nice, no?
So let's see, students. So it is all. It's so nostalgic to me. All of this. I studied this like years back, and everything is coming back to me. And it's always fun to learn chemistry. I don't know why. It's always been fun to learn chemistry and all this.
For me, bio is also fun to learn, but the problem is the names. If you say that, ma'am, you don't have to remember the names, I'll tell you the process. Then I
But-
will like, sir, I can
But
sit
the
there.
process will involve the names.
Yeah, it's like more of history, no?
Huh.
In history, you like history.
Huh.
Like it's very interesting.
But you
But
don't
I
like
don't
dates.
want.
You
Yeah,
don't like
I don't
dates.
want to
You
learn
don't
the
like
dates.
dates.
Just tell me the process.
Okay, last question for you, ma'am, students. Students, for all of you also, last question. Let's go if you're able to solve this question. And we are enjoying today. This was a very fun session. We thought, so Diksha ma'am was in Bangalore. We decided let's have a fun session together where we can interact with students and learn something new. And obviously, I'm also learning something new.
Yeah,
So,
and I'm learning so much
Huh.
about
Yeah.
plants.
ma'am, I've told this to many students of my DNA batch also. There is certain fungi which can mind control.
Really?
It is called as zombie fungus, cordyceps. It can, this fungus spores will be produced. A poor ant will take the spore, it will breathe the spore will develop in the ant's brain. Fungus will grow in the ant's brain, it will release chemicals and control the ant and make sure the ant goes to suicide. So plants.
Is it applicable only to ants or?
That is the first question when I had. So fortunately, it is only for ants. It cannot attack us and all. So it's called as cordyceps.
So basically, the movies that are made or zombies, that is basically an elaborated or exaggerated
Exactly, and it's
version.
real. It's real. This is real. It's called as zombie fungus.
So if that zombie fungus somehow attacks human beings, then we'll get
No,
converted.
because our immune system will break down the spores. It will not allow.
Somehow, then those movies that we see on
It may become real.
Okay. Now
So,
that was such an
see,
interesting thing.
so plants are amazing, no?
Yeah, plants are amazing. Yeah, that's why we are living.
Ma'am, technically speaking, fungus is not a part of plants actually. because there is something called as five kingdom classification. We have your plants and fungus are separate. Protista, Monera, Plantae, Animalia, and
studied this.
Diversity. Diversity, exactly. Okay, ma'am.
Class we have.
Now, yes, in 9th class we have. Oh, I remember everything.
Yeah, word to word, I remember mammals, reptiles. We are mammals, reptiles, lizards, and all.
Yeah,
Right?
then
So,
apes.
to word. I don't remember that much.
Okay, ma'am. Next question for you. Difficult question coming up. Hot or difficult question?
Why you are so obsessed with enzymes? So every question is related to enzyme. So there is activation energy here, so I feel that I can answer this.
Okay. Right.
Activation
It's
energy.
easy,
है.
it's easy question. It's easy question.
In a reaction catalyzed by an enzyme, which of the following statement is correct? Enzyme decrease the activation energy for the formation of transition state. No. We are talking about promoters here, right? Enzymes make transition from substrate to product more difficult. No. Enzymes increases the activation energy. Wait a minute. Erase that. Huh. Wait a minute. That is definitely wrong. Enzymes decreases the activation energy to, so it will decrease. It is like this now. So it will decrease the activation energy. It will make the process easier. So that is true. Decreases, that is wrong. Enzymes complex formed in reaction lasts for a very, no, it is very short time.
See, such an easy question.
Huh.
Such an easy question. Did you realize how did ma'am broke down the entire thing? Right? So easily she broke down the concept. Now, oh ma'am, I'll give you a simple example. Okay. Now, have you ever tried high jump? No.
Do I look like
Okay. Imagine ma'am is doing high jump. Okay. For example, you're running and coming. I will keep a bar here. And you're supposed to jump over this bar. Can you do this?
tried it.
Okay, this much now.
Failed.
This much. Can you jump this much height?
I used to do this.
Okay, this much height.
No, that I can't do. I'm just saying that I used to do high jump.
High jump. Okay, ma'am.
But
So.
I used to fail in that. That's what I'm doing. This much jump.
Okay, so
in a-
ma'am, I said
in
we
a
can
elephant-
clear the jump. I didn't say participate. I mean clear the
So
jump.
I'll tell you one thing. In our school annual functions, it was, first barrier was this much, this much. Then it used to increase. Next step was this. Third step was used. So at one point, I'm sharing this on camera. So there was so much soil here. Okay, so I used to run like, okay, I was very competitive in nature. So I was like, I will crack this. I will crack this, and it's like physics will be used here. My speed was too fast.
So you were under it.
I jumped it, and I jumped it to that extent. That was such a high jump that my next barrier, I almost stepped onto that, and my nose was almost like this. I was like, relax.
Enough,
Isha.
ma'am. Enough, ma'am.
Relax.
Enough.
Like, okay. And after that, I had this phobia. I was like, I will never ever do in my life. I've
High
never
jump.
do. No, I've never done that. I was like, I'll break my nose or my teeth or something. I'll do.
Okay, ma'am. Why am I imagining you breaking your teeth now?
No, they are intact. Don't
Okay.
worry.
So, ma'am, okay. For example, you're supposed to come and jump over this barrier. Will you be able to do it? Difficult, no? Like, will you be able to come jump over this height?
No.
Okay. Now, so this is the activation energy right now. Now, what will enzyme come and do? Enzyme will come and decrease the activity till here. Can you jump now?
Yeah.
That's all.
Yeah. So this is also very
In chemistry also, right? Yeah, it'll be there in
We have
chemistry.
proper in chemical kinetics as well. In
Oh, yeah,
chemical
there is
kinetics.
Chemical kinetics, obviously you learned
Yeah,
that.
that's what I was telling you.
This is the activation energy with enzyme, without enzyme. Enzyme is decreasing the activation energy.
So basically, this is more of a concept of chemistry because transition state also I taught you in whether it's organic chemistry, we teach them about intermediate transition states,
state.
right? So transition state is so very, very unstable. Intermediate is still like
Fine.
little bit,
Right?
but this is very
unstable, so
unstable.
energy will be very high. Very high energy, so energy will decrease the energy, right?
Yeah. So basically, also one more thing here. Am I right or wrong? Whenever in catalyst, so I tell my students that if nothing is mentioned to you, they are talking about positive catalyst.
Positive, yes, always.
Enzymes also it is
Yeah,
true.
yeah, yeah. So enzymes, we usually talk about positive catalyst only. There is something called negative enzymes. Usually enzymes will hasten the reaction always.
Inhibitor also.
Inhibitors we usually don't talk about here. Inhibitors will stop the reaction, that's all. Don't slow down. Stop it.
Okay, it will stop only the reaction. For example, some person
Competitive,
has
uncompetitive, and non-competitive inhibitors.
not that much detail. I have read it somewhere, but not that much in detail.
So students, I think that is the end of today's class, ma'am.
I wish we can.
We could solve more questions, but more questions are not there for today. So let me know in this. Let me know in the comment section today's video. How was today's session? Did you have fun? And it was a very minor interaction with all of you, telling that importance of chemistry and biology in biochemistry,
Right.
where it's a two chapters where two different concepts are coming together. You're learning something new: chemistry aspect, bio aspect. So.
So
ma'am,
yes.
anything to the students?
So how much marks you will get out of these?
So
biomolecules?
biomolecules usually they ask around 4 to 5 questions.
Really?
Yeah,
4
yeah,
to 5
4 to 5
questions.
questions.
Wow, amazing. I was expecting 2 to 3 questions.
No, 20 marks easily. 20 marks
Really?
easily 20 marks.
From just bio only.
Biomolecules.
Biomolecules.
Hmm.
From chemistry, it won't be 4 to 5, but yeah, 2 to 3 questions they
2 to
can
3
ask
questions.
because there are naming reactions also.
Okay.
But yes, they will ask you more of structure based questions in chemistry
Okay.
rather than theory based questions. But yeah,
Makes
I
sense.
think if you have done, if you have done biomolecules and chemistry biomolecules together, you can easily score around 7 to 8 questions from there.
Exactly,
Right?
7 to 8 questions.
So I think that is amazing that you are
Yeah.
getting from one chapter, from one concept, you are getting these many marks.
Exactly.
Right?
So,
Diksha sir, they are
Diksha
telling
sir
in the
again. Diksha sir and Diksha ma'am together.
chat. That's epic, guys. That's
Oh
epic.
god. Always. That joke never ends.
Yeah.
Okay, students, thank you so much for today's class. Quickly smash that like button and let us know in the comment section, because if you show the love, we might consider doing such classes more and more. It's always fun to do with all of you. So thank you so much, students, and we will see you in the
One more thing here. So, guys, we are planning, and I want all of you to write it down in the comment section. We are planning me and Shreya sir, because so chemistry is everywhere. Sir. Excuse me. So in bio also it's chemistry. Chemistry is like I'll be friends with bio also. In physics also we have two chapters that are common. One is
So
thermodynamics.
why don't you and sir do in class then?
We were thinking about that, right? So I want all of them. I want
Okay.
you guys to comment down below that what you want, right? We have two chapters, thermodynamics, and we have structure of atom. There are so many concepts that are exactly there in physics and exactly there in chemistry.
So you can do it combined
Yeah,
class.
it would be great. Okay.
Crazy then.
So guys,
Thank you so much.
D for Diamond Dosa Man, B for Brilliant Baswa sir. Finally, you have your name back. Congratulations.
So
Okay, then bye bye, guys. See
Thank
you
you so
all
much.
in the next class, next
session, next session. So bye bye, take care all of you. And students, those of you who are not in MVSAT exam, quickly go check it out. The MVSAT, it's still on. Link is in the description. Go check it out once. And thank you so much for today's class. Bye bye. Take care all of you.